Yan. Okay, so our topic for this morning is porphyrins, RBC, and respiration. Again, shown there is the link to my Telegram account so that if you have questions, clarification, you can send me a direct message there. Right, so here are our learning objectives. I will not go through them one by one as they are already written in your lecture guide. Let's discuss the first two. What is porphyrin? So porphyrin is a cyclic compound, kita nyo naman sa structure, made up of 4 pyrrole rings connected through methine bridges, complex with metal ion, and depending on the metal ion it will complex with, that will determine what type of porphyrin it will be. In humans, it's commonly complex or associated with iron so that we form your heme. Okay? On the picture, you'll see that porphyrin can be seen in other molecules in our environment. For example, in the chlorophyll of plants, you can see a porphyrin with magnesium complex with it. On your left, that is a vitamin, your cyanocobalamin complex with cobalt ion. Okay? So the numbering of your porphyrin rings, the ring itself uses Roman numerals 1 to 4, or in other textbooks, in other sources, it's using English alphabet, A to D. It's numbered from top going in a clockwise direction. 1, 2, 3, 4. A, B, C, D. The methine bridges, the one that connects your pyrrole ring, is labeled with Greek alphabets. Clockwise din po ang labeling niyan. So it's alpha, beta, gamma, and delta. Again, proceeding in a clockwise direction. Clear po? Very important na maalala yung structure because later on, when we discuss the synthesis and degradation, makikita niyo po yung importance ng structure. Excuse me. All right. So the porphyrin nucleus are numbered, or are labeled, the carbons, the peripheral carbons are labeled 1 to 20. So yung nasa periphery are carbon atoms, And the inner nitrogen atoms, are given locants 21 to 24. Porphyrin, can have side chains. Pwede mong kabitan ng side chains in 2 positions. One is in the beta position, located on the pyrrolic rings, di ba? Ito yung ring natin. Or in the meso position, located on the methine bridges, 5, 10, 15, 20. In humans, the substitutes, or the side chains are commonly attached in the beta position. Okay? And a porphyrin, that does not have any side chain attached to it, walang kahit anong side chain, is called a porphin, the simplest porphyrin. Next, as I've mentioned, you can attach side chains to your porphyrin, such that the common side chains are as follows: acetate or acetic acid, otherwise known as carboxymethyl, propionate or propionic acid, or carboxyethyl. These 2 side chains are the polar side chain because of the presence of carboxyl group. Okay? While methyl and vinyl are the non-polar side chains. Nasundan po? On your right, you'll see the common porphyrins and porphyrinogen we see in humans. First is your uroporphyrinogen, containing 4 acetate and 4 propionate side chains. Coproporphyrinogen with 4 propionate and 4 methyl side chains. Protoporphyrin 9 or protoporphyrin 3 with 4 methyl, 2 vinyl, and 2 propionate side chains. Okay? Tandaan yung mga side chains. That's very important when we discuss heme synthesis. So the nature and distribution of the side chain determines its property, no? More commonly, its solubility, such that presence of carboxyl group, presence of polar side chain makes the porphyrin water soluble. So if I ask you between uroporphyrinogen and coproporphyrinogen, which is more polar or more water soluble? Sino po ang mas polar sa kanila? Tan, Cesar, Anthony? Napabukas ng camera si Lian. Anthony, andito ka? Between uroporphyrinogen and coproporphyrinogen, which is more water soluble? Sige, tawag tayo ng iba. Gideon, Restore. Mr. Restore?
Hello, Doc.
Hi.
Yung... Ay, sorry. Maingay. Yung uroporphyrinogen po.
Because?
Yung side chain niya po makes it more polar because it's carboxymethyl.
Okay, because of the acetic acid and propionic acid side chains. Okay? But between coproporphyrinogen and protoporphyrin or protoporphyrinogen, which is more polar, si coproporphyrinogen na po. Kasi ang protoporphyrin, meron na siyang vinyl side chain that are non-polar. Alright. So we can classify porphyrins into 4 types, no? But in the interest of medical biochemistry, we'll only be discussing the type 3 porphyrin, no? We can identify the type 3 porphyrin by the presence of an asymmetric side chain on ring D or ring 4. Parehas din po yun. Diba sabi natin, the pyrulic rings can be labeled using Roman alphabets or Roman numbers, and English or English alphabet. Such that in ring D sa type 3 porphyrin, you have an asymmetric side chain. What does that mean? Take a look at the ring A. It's acetate, propionate. Ring B, acetate, propionate. Acetate, propionate. Pero pagdating sa ring D or ring 4, it's interchange, propionate, acetate. Yun po yung tinatawag nating asymmetric side chain. Nasundan po? Okay. So we're now done with porphyrin. Let's now discuss heme. What is heme? So heme is a flat planar structure containing porphyrin ring with iron on its center. Because of that iron, it allows the heme to transport electrons, kasi yung iron niya can go oxidation and reduction, So pwede siyang magdala ng electrons, like oxygen. And usually, most of the time, heme is not present in its free form. Hindi siya present in our body as free form, kadalasan. They are usually associated with proteins. Ang tawag natin doon with heme attached to protein, that's heme proteins. And depending on the protein where it will associate with, kung saan siya kakabit, that will determine the function of heme protein. For example, hemoglobin. Hemoglobin has 4 heme molecule attached to 4 globin chain. Therefore, it is used for transport of oxygen. Myoglobin has a single globin chain attached to heme. Therefore, it's only effective in storing oxygen. Cytochrome C, saan nyo na-encounter yung cytochrome C?
Electron transport chain po, Doc.
Correct, no? It's one of the mobile elements of your electron transport chain, di ba? So, kinakabitan din ba yan ng electrons? Yes po. Sabi nga natin, that is because of the iron present on it. Cytochrome P450 is an enzyme produced by the liver needed for hydroxylation of xenobiotics. Doc, ano po ba yung xenobiotics, no? So, xenobiotics are endogenous or exogenously synthesized compound that are lipophilic, no? They are lipophilic. For us to excrete them, we need to convert them to polar compounds or water-soluble compounds. So this enzyme, would hydroxylate it. Pag kinabitan po yan ng hydroxyl group, it will make the molecule polar that can be excreted. Next catalyst, you've already encountered this. No, this is responsible for the degradation of hydrogen peroxide to water and oxygen, and tryptophan pyrrolase for oxidation of tryptophan. Any questions with porphyrin and heme, the structure? Guys, may question po? Heme and porphyrin. So I'll take that as a no. Let's proceed with heme synthesis. So heme synthesis, no, is synthesized at a rate, no, of 0.3 grams per day. There are two major organs that carries out, no, heme synthesis. First is your bone marrow, specifically in the erythrocyte precursor cell, no? Majority of heme synthesis occurs there, and the rest is in the liver, no, where it synthesizes heme, no, as part of heme-containing enzymes, no, like cytochrome P450, cytochrome C, and other enzymes. This pathway uses two cellular compartments, no. You have your mitochondria as well as the cytoplasm. So it's one of the few pathways in our body that utilizes both cytoplasm and mitochondria. The other pathway, one pathway is in protein metabolism. You've already discussed it. Ano yung pathway na yon that uses both the mitochondria and cytoplasm in protein metabolism? Starts with the letter U.
Urea cycle po.
Correct. Urea cycle. The other one, you'll take up, no? Sa carbohydrate metab, that's gluconeogenesis. Okay? Gluconeogenesis. And our topic for today, no, is heme synthesis. So when I was reviewing for the board exam, our mnemonic here is that hug, no? Yung cytoplasm and mitochondria seems to be hugging each other, no? So this pathways, no, is seen in these two cellular organelle: heme synthesis, urea cycle, and gluconeogenesis. Okay? So this is just an overview of your heme synthetic pathway. As I've mentioned, it occurs at two cellular compartments, the mitochondria and the cytoplasm. The initial step happens in the mitochondria. The intervening step happens in the cytosol. And the terminal 3 steps goes back to the mitochondria. All right? So with that, let's discuss each step. First is formation of delta aminolevolinate or delta aminolevolinic acid, in short, ALA. Succinyl-CoA that comes from the Krebs cycle. No? Ang succinyl-CoA, it's actually an intermediate of Krebs cycle or TCA cycle. That should give you an idea that this pathway happens in the mitochondria. Kasi ang Krebs cycle is in the mitochondria. So the succinyl-CoA, that is a substrate for the first enzyme, is present here. Will condense with glycine to form D-ALA or delta aminolevolinic acid. The enzyme needed, ALA synthase, which is the rate-limiting enzyme. Guys, pag nag-aaral kayo ng mga pathways sa biochemistry, tatandaan nyo, kailangang maalala ang rate-limiting enzyme, no? Yan yung mga isa sa control point, no? Must know yan. Dapat in-snap. Alam natin yan. So it's a decarboxylating enzyme, kaya makikita nyo may release ng coenzyme A and carbon dioxide. This enzyme requires, no, a coenzyme in the form of pyridoxal phosphate that is derived from vitamin B6. Okay? So you'll not be surprised that patients with vitamin B6 deficiency, can they develop anemia? Pwede po, no? Kasi pyridoxal phosphate, which is the coenzyme for ALAS, is needed for you to synthesize heme. And heme is needed for hemoglobin synthesis. Okay? Next, there are two isoforms of the enzyme, ALAS1 and ALAS2. ALAS1 is also called as the housekeeping enzyme. Okay? Encoded in chromosome 3. ALAS2 is specific for erythrocyte precursor cells. It is encoded in the X chromosome. Okay? In the X chromosome. Presence of heme as well as glucose would inhibit ALAS, and that presence of iron, which is found at the center of your heme, is a positive regulator of the enzyme, meaning it will stimulate the enzyme. Please take note that heme is the end product of the pathway. Tama po ba? Therefore, heme acts as a negative feedback inhibitor of ALAS. Nasundan po? Clear so far? Okay. Next, 2 molecules of ALA would go outside of the cytoplasm. Sabi natin, di ba, yung first step lang sa mitochondria, the intervening steps are in the cytoplasm. So 2 molecules of ALA would condense the first precursor pyrrole, your porphobilinogen. Sabi natin, di ba, dun sa molecule ng porphyrin, how many pyrrole ring does it have? Ilan? Naalala nyo kanina, pinakaunang slide, 4 pyrrole ring. So gagawin natin ngayon yung first precursor pyrrole. Okay? That is with the help of the enzyme ALA dehydratase or ALA dehydrogenase. This is a zinc requiring enzyme. Kailangan niya ng zinc. So that presence of heavy metals like lead, presence of heavy metal like lead can actually chelate the zinc and render the enzyme inactive. Okay? Next, 4 molecules of porphobilinogen. Kasi nga po sabi natin, ang porphyrin has 4 pyrrole ring, would condense to form the linear tetrapyrrole, otherwise called as your hydroxymethylbilane, catalyzed by the enzyme porphobilinogen deaminase, otherwise called as your hydroxymethylbilane synthase. What can you notice about the side chain? It contains acetate and propionate side chain. Hydroxymethylbilane can undergo spontaneous cyclization or non-enzymatic cyclization to form the type 1 uroporphyrinogen, or enzymatically with the use of your uroporphyrinogen 3 synthase to form the type 3 uroporphyrinogen. Again, how do we distinguish type 3 porphyrinogens or porphyrins from other porphyrinogens or porphyrin? That is the presence of an asymmetric side chain on ring D or ring 4. Acetate propionate, acetate propionate, acetate propionate, propionate, acetate. Nagbaliktad po. Between the two, which is the physiologic type of porphyrin, it's your type 3 uroporphyrinogen or uroporphyrin. Okay? Next, uroporphyrinogen, this is still in the cytoplasm, would undergo decarboxylation with the help of the enzyme uroporphyrinogen decarboxylase to form coproporphyrinogen type 3, converting the acetate side chains to acetate side chain, to methyl side chain. Again, natutunan natin kanina, this will decrease the polarity of your porphyrin. So pag tinanong kayo, between the two, sino ang mas ma-isolate natin sa urine? We can actually check the porphyrins in urine or in the stool. So sa urine, between the two, sino ang mas ma-isolate natin? It's your uroporphyrinogen or porphyrin. Kaya nga ang pangalan niyan, may uro sa pangalan, no? Kasi it's initially isolated in the urine. Alright? So this picture just shows you that again, acetate side chains are converted to methyl side chain. All right? Now, coproporphyrinogen would go back to the mitochondria to undergo series of oxidation, initially from coproporphyrinogen to protoporphyrinogen 9 or protoporphyrinogen 3 with the help of the enzyme coproporphyrinogen oxidase, converting 2 propionate side chains to vinyl side chains, such that the side chains you see in your protoporphyrinogen 9 or protoporphyrinogen 3 are 2 vinyl side chains, yung propionate na bawasan ng 2, 2 propionate side chains, and how many methyl side chains? 4. Wala nang acetate side chains. So tinanong kayo, between the three, if you arrange it from polar to least polar, most polar, uroporphyrinogen, followed by coproporphyrinogen, and the least polar is your protoporphyrinogen. Another round of oxidation converts protoporphyrinogen to protoporphyrin with the help of the enzyme PPOX, protoporphyrinogen oxidase. What can you notice about the difference of protoporphyrinogen and protoporphyrin with regards to its structure? Especially in the bridges that connects your porphyrin rings or the pyrrole ring. So makikita nyo, from single bond, naging double bond. So yan yung makikita natin sa porphyrins, presence of double bond. So porphyrins is the oxidized form of your porphyrinogens. And because of this double bond, this allowed the molecule to absorb light, no, and display color. This is the reason why our blood is red, no, because of this specific structure. Okay? Please remember that also because of this double bond, when porphyrins are exposed to UV, they will generate reactive oxygen species. Bad po ba yung ROS? Bad po ba yung reactive oxygen species? Yes po, because it can damage the membrane of our cell, no? Kasi nga, nagsascavenge siya ng electrons for it to be stable. So remember that. Also, porphyrinogens can undergo spontaneous oxidation to their porphyrin counterpart even without the use of enzyme. Tatandaan niyo yun. So that uroporphyrinogen can oxidize to their uroporphyrin counterpart kahit walang enzyme. Coproporphyrinogen can undergo spontaneous oxidation to their porphyrin counterpart even without the use of enzyme. Later on, makikita niyo yung importance niyan when we discuss porphyria. Basta itanim niyo lang sa isip niyo that they can undergo spontaneous oxidation. All right? So this table just shows you the difference between porphyrins and porphyrinogens. They're pretty much self-explanatory. No? So wag na po natin isa-isahin. The last step, still in the mitochondria, would be the incorporation of iron to your protoporphyrin 9 using the enzyme ferrochelatase. So we now form your heme. Heme can then go out of the mitochondria, and again, it can associate with proteins, di ba? Sabi natin, to form heme protein. And depending on the structure of the protein it will associate it will determine its function. So pag yan, hemoglobin. Pag dito, myoglobin. Okay? Please remember that iron, as you can see here, I've already emphasized this a while ago, is a positive regulator of ALAS, meaning it stimulates ALAS production. Heme, the end product of the pathway, sabi natin, is inhibitory or is the negative regulator of ALAS. Okay? Ferrochelatase, just like ALA dehydratase, is a zinc requiring enzyme and presence of heavy metal, no? Like lead, can also inhibit ferrochelatase. Nasundan po? With that, let's just briefly go back and review everything that we've discussed. Succinyl-CoA A would condense with glycine in the mitochondria to form your D-ALA with the help of the enzyme ALA synthase, the rate-limiting enzyme, requiring pyridoxal phosphate derived from vitamin B6. Two molecules of ALA would go outside of the mitochondria to form the first precursor pyrrole, porphobilinogen, with the help of the enzyme ALA dehydratase or ALA dehydrogenase. Four molecules of porphobilinogen would condense to form your hydroxymethylbilane or your linear tetrapyrrole with the help of the enzyme PBG deaminase, otherwise known as your hydroxymethylbilane synthase, with the side chains acetate propionate. For acetate, for propionate side chain. It can undergo spontaneous cyclization to form the type 1 uroporphyrinogen or enzymatically using uroporphyrinogen 3 synthase to form your uroporphyrinogen 3. Again, still with acetate and propionate side chain. Okay? Uroporphyrinogen would undergo decarboxylation to form coproporphyrinogen, converting the acetate side chains to methyl side chains. Coproporphyrinogen would go back to the mitochondria to undergo series of oxidation, initially to your protoporphyrinogen, converting some of the propionate side chain to vinyl side chain, methyl 4. Okay? And protoporphyrinogen 9 would oxidize to protoporphyrin 9, eventually addition of iron with the use of ferrochelatase to form now your heme. Heme can go outside of the mitochondria to associate itself with proteins, and depending on the protein it will associate it with, it will determine its function. In this case, it's your hemoglobin. Any questions up until this point? Clear po? All right. So let's now proceed with the next objective. So guys, pag mag-aaral kayo sa biochem para dumali ang buhay nyo, no? Hindi po lang pinag-aaralan yung pathway. Siyempre, magiging doktor din po kayo, di ba? Yes, doc, kine-claim ko na po yan. Magiging MD po ako, magiging licensed doctor po ako. Pag naging licensed doctor ba kayo, i-examine lang ba kayo sa mga pathways na alam nyo? Hindi po. You'll actually apply it in real-life patients. So dapat, alam nyo din po ang mga diseases related sa pinag-aaralan nating pathway. Tama po? Yes po. In the case of heme synthesis, no, the diseases related to it is your porphyria or porphyrias. Okay? So let's now discuss porphyrias. Kilala niyo po yung tao na nasa picture? Sino yan? US President na may ganyang hat.
Abraham Lincoln, Doc.
Okay, si Abraham Lincoln. Napanood niyo yung movie niya na ano siya, Vampire Slayer siya?
Yes po, Doc.
Yes, no, di ba? Maganda yun, no? So sabi nila, no, porphyrias daw, that is the medical explanation daw for vampires, no? Bakit? Ang vampires ba, takot sa araw? Yes po, Doc. Kasi sabi nila, some patients with porphyrias would present with photosensitivity. Ang mga vampires ba, mapuputla ba sila? Yes po, Doc. Kasi they have, sabi nila, ang porphyrias daw have defective heme synthesis. Di sila makagawa ng heme. Di tuloy sila makagawa ng hemoglobin for RBC production. Ang mga vampires ba, ano ba, weird ba yung kanilang mga behavior? Pumapatay ba sila? Yes. Because some patients with porphyria would present with neuropsychiatric symptoms, no? Neurovisceral symptoms, no? Pwede silang may depression, pwedeng psychosis, no? Kaya odd daw yung behavior nila. So, ano ba yung porphyrias? So, these are rare metabolic disorders caused by altered activities in any of the enzyme of your heme synthetic pathway. And we can categorize it into two based on the organ it primarily affects. It can be hepatic or erythropoietic. Okay? Sa liver or sa RBC. And there are two symptoms, no, or two clinical manifestation that you can observe in porphyrias. One is neurovisceral symptom presenting with abdominal pain, motor and sensory peripheral neuropathy, or neuropsychiatric changes, no? Yun nga, sinasabi ko, depression, psychosis, no? Iba-iba, behavioral problems. Or cutaneous manifestation that can either be chronic and blistering or acute and mostly non-blistering. Please take note, very important po to. Neurovisceral manifestation is caused by elevation in the earlier intermediates of heme synthetic pathway like ALA and PBG. Nauna po ba natin ginawa yung ALA and PBG dun sa heme synthetic pathway? Yes po, Doc. First and second step. ALA, delta aminolevulinic acid or porphobilinogen. That is because ALA and PBG are said to be damaging to the neurons. Kaya ang symptom nila pag yung dalawang nag-elevate is neurovisceral symptom. Cutaneous manifestation like this patient with porphyria cutanea tarda is due to elevation in the porphyrinogens spontaneously oxidizing to their porphyrin counterpart. Pag yung structure ng porphyrin, circular na siya, di ba? Pag bilog na siya at naging porphyrin siya, di ba sabi natin, because of that double bond, pag na-expose siya sa UV rays, ano ang ma-generate natin? Reactive oxygen species. So that when it circulates on the top portion of the skin, it can damage the skin, causing now the photosensitivity we see in porphyrias. Nasusundan po? Clear so far? Sige, yan po ay very important kasi di-discuss natin yan later. So again, just to emphasize, no, mutations in various genes, no, in your heme synthetic pathway is the reason behind porphyria. Accumulation of ALA and PBG, ano uling manifestation? Neurovisceral or neuropsychiatric symptom. Accumulation of porphyrinogens. Pwede ba silang mag-spontaneously oxidize to their porphyrin counterpart? Yes po, no? That will be the one causing photosensitivity or cutaneous manifestation. Okay? So with that, pwede na po nating i-discuss ang different types of porphyria. Dadaanan lang po natin to, no? Kasi alam nyo naman na pag ang nag-elevate ay ALA and PBG, again, what's the manifestation? Neurovisceral. Pag porphyrinogens, spontaneously oxidizing to their porphyrin counterparts, cutaneous manifestation. Or pwede rin namang combination of both, neurovisceral and cutaneous. So dapat memorize nyo na yung pathway, no? Hindi ko naman sinasabing memorize nyo na ngayon, no? Pag nag-aral kayo. Kasi kapag alam nyo which step or which enzyme is affected, the intermediate before it is expected to elevate. Tama ba? So kapag alam nyo yung mag-elevate na intermediate, alam nyo na yung manifestation ng patient. Alright. So let's start. X-linked protoporphyria. In X-linked protoporphyria, this is a gain of function mutation in ALAS2. Di ba sabi natin kanina may dalawang isoform ang ALAS, ALAS1, ALAS2. ALAS2 is specific for the is specifically encoded in the X chromosome in the erythrocyte precursor cells. Tama po ba? So the organ primarily affected are your premature RBCs. Excuse me. So anong problem dito? Overly active yung ALAS. G na G siya, no? So kung G na G yung ALAS, tataas ba yung intermediates lahat? Yes po, Doc. Tataas ba ang ALA and PBG? Ano pong manifestation pag ALA-PBG? Doc, peripheral neuropathy, neurovisceral symptoms. Eh yung mga porphyrins po, pag nag-elevate, cutaneous symptoms, no? Expected naman na all porphyrias will have anemia, no? So mag-focus lang tayo doon sa symptoms related to it. Next, aldehyde porphyria. This is also known as plumboporphyria or DOS porphyria. This is the rarest of all your porphyria. I think meron lang 2 or 3 diagnosed case nito. Worldwide na yan, ha? So ang problem here is in the second enzyme, ALA dehydratase, ALA dehydrogenase. So the intermediate before it would also elevate. So ano expected nating manifestation ng patient? Neurovisceral or cutaneous? Neurovisceral. But please take note, there's always an exception to the rule so that this patient, no, based from literature, can also have elevation in your coproporphyrin. But also please take note that this is not clinically significant. It is not clinically significant, therefore, ang manifestation lang po niya is purely neurovisceral. Okay? So, there are conditions that can actually mimic ALA dehydratase porphyria. Yung symptom niya, similar. Pero ang problem, hindi genetic that causes deficiency of the enzyme. First, lead poisoning. Nakita na natin to kanina. Sabi natin, ALA dehydratase, is it a zinc-requiring enzyme? Yes po. Kailangan po niya yung zinc for its activity. Therefore, presence of lead can chelate that zinc, no, rendering the enzyme inactive. Next, hereditary tyrosinemia type 1. Nadaanan na po ba natin to? Na-discuss na po ba to? Hindi pa. Naku, patay ka, Miss Desiderio, kay Dr. Santos, no? Protein metabolism, hereditary tyrosinemia type 1, no? Is a defect in metabolism of your tyrosine. So anong problem dito? Bakit niya imimimic yung alad dehydratase porphyria? Ito po. In hereditary tyrosinemia type 1, you have a problem converting fumarate acetoacetate to fumarate and acetoacetate. The enzyme needed there, please remember, fumarate acetoacetate hydrolase. So if you cannot convert fumarate acetoacetate to fumarate and acetoacetate, it will be shunted, no, to an alternative pathway leading to an increased level of succinyl acetone. Succinyl acetone, as you can see on the last bullet, no, is the most potent inhibitor of ALAD. So in that sense, mamimimik niya ba yung symptoms ng ALAD dehydratase porphyria? Yes po, Doc. Okay? Next, acute intermittent porphyria. Acute intermittent porphyria is the most common type of porphyria presenting with neurovisceral symptom. The problem here is in your porphobilinogen deaminase or hydroxymethylbilin synthase. So that what are the intermediates before it? ALA and PBG. Anong manifestation? Neurovisceral or cutaneous? Neurovisceral. Okay? Next, congenital erythropoietic porphyria. The problem naman here is in your uroporphyrinogen 3 synthase. Manifestation of the patient is photosensitivity and hemolytic anemia. Question, bakit po magpe-present ng photosensitivity kapag may inhibition or deficient uroporphyrinogen 3 oxidase? Doc, hindi nyo pa naman nagagawa yung mga circular porphyrinogens that can spontaneously oxidize to porphyrins. Yung pong mga before it, no, are the one causing neurovisceral symptom. Bakit po photosensitivity yung symptom ng patient? Tingin nyo, guys.
Non-enzymatic conversion.
Correct, no? Mama Eric, no? That is because, no, once you form hydroxymethylbilin, di man po siya makadaan dun sa enzymatic conversion, pupunta siya sa non-enzymatic cyclization, forming your type 1 uroporphyrinogen and type 1 coproporphyrinogen. Can that also spontaneously oxidize to their porphyrin counterparts? Yes po. Can they cause photosensitivity? Yes po. Next, porphyria cutanea tarda. Among all the porphyrias, this is the most common. Ito po ang pinaka-common, no? So this is due to the deficiency in your UROD enzyme, uroporphyrinogen decarboxylase. So ang manifestation ng patient, there will be elevation in your uroporphyrins. So ang manifestation is photosensitivity, no, or cutaneous symptoms. Hence the name, porphyria cutanea tarda. Please remember that you'll only present with this condition once the enzyme activity is less than 20%, no? Very efficient po ang uroporphyrinogen decarboxylase. Kapag 30, 40%, no, still you will not present. Only when less than 20%. Next, let's discuss naman the different types of mixed type of porphyria. First is your variegate porphyria. No? The defect here is in your PPOX enzyme, protoporphyrinogen oxidase. So anong manifestation nito? You'll have elevation in ALA, PBG as well as your protoporphyrinogen. No? So anong manifestation? Neurovisceral, cutaneous, or both? Both. Okay? Doc, sabi mo, pag yung protoporphyrin, hindi naman natin ma-convert yung protoporphyrinogen to protoporphyrin kasi may enzymatic defect. Bakit pa rin magkakaroon ng increase in protoporphyrin 9 at photosensitivity?
Spontaneous oxidation.
Correct. No, that is because protoporphyrinogen can undergo spontaneous oxidation. Please remember that presence of protoporphyrinogen can stimulate ALAS and at the same time inhibit porphobilinogen deaminase, furtherly increasing ALA and PBG. So manifestation is mixed type porphyria. Next, hereditary coproporphyria. Same lang din po. It's a mixed type of porphyria with elevation of ALA and PBG, but this time coproporphyrin na po ang mag-elevate. Okay? So, pero ganun din po ang manifestation. Neurovisceral, cutaneous manifestation. And lastly, your erythropoietic protoporphyria. This one naman is the defect in the terminal enzyme, ferrochelatase. So, ang manifestation ng patients na to are cutaneous symptoms. Okay? So, primarily affecting the RBC. Okay? Okay. Mali pala, that pala yung next slide ko. So, meron silang anemia because they cannot synthesize heme. All right, so that's it for porphyrias. Let's now discuss alcohol and other CYP 450 inducers. In exams, no, sa exam, and in clinics, no, pag naging doctors na kayo, patients with porphyria would usually come to the emergency department, no, or to your clinic with attacks of porphyria after binge drinking, after intake of cytochrome P450 inducers. Okay? Tatandaan niyo yan. Sige. So, bakit? Okay. So, tingnan muna natin yung passage. The degree of overproduction of the intermediates of heme synthetic pathway. Ano nga ulit yung mga intermediates? ALA, PBG, ALA, PBG, the porphyrinogens spontaneously oxidizing to porphyrins, di ba? The degree of overproduction of those intermediates is dependent on the stimulation of ALAS. Pag stimulated po ba yung ALAS, madami yung intermediates? Yes po, Doc. Doon nga sa first type of porphyria, X-linked protoporphyria, GC ALAS. Kaya po yung symptoms, kita natin lahat. Such that, makikita nyo dito sa baba, no, on the table, are the positive and negative regulator of ALAS. We discussed na po already yung iron. Tama po ba? It's a positive regulator of ALAS. It stimulates ALAS. So ang patient nyo kapag may porphyria, lumaklak ng iron, magmamanifest ba yan ng porphyria or ma-aggravate ba ang symptom niya? Yes po, kasi stimulatory po siya sa ALAS. Alcohol, sabi natin, and CYP 450 inducers can also stimulate ALAS. How? So first, ethanol. Paano ba natin na-metabolize yung ethanol? Ethanol at lower concentration is metabolized by two enzymes, alcohol dehydrogenase to acetaldehyde and acetaldehyde dehydrogenase to acetate. Pero kapag ang alcohol is very high in concentration, nag-binge drinking after midterm exam, nakita yung result ng exam, naglaklak, meron pala siyang porphyria, other enzymes would help, no, in the metabolism of ethanol. One that is not shown here is catalase. Pwede rin pong i-degrade ng catalase ang ethanol. But what's more important in our discussion is your cytochrome P2E1. Guys, tatandaan nyo, cytochrome P2E1 is a member of the cytochrome P450 family. Kapamilya po siya ng cytochrome P450. Ang cytochrome P450 po ba is a heme protein? Yes po, Doc. Therefore, may heme ba siya sa structure niya? Yes po, Doc. Ngayon, kakagamit mo ng heme for you to synthesize cytochrome P2E1 para lang may metabolize mo yung ethanol. What would happen to the level of your heme? High or low? Bababa, gamit ka ng gamit. And sabi natin, ano nga ang function ng heme? That is to inhibit ALAS. Ngayon na mababa na ang heme, gina ba uli si ALAS? Yes po, Doc. No? In that sense, mape-precipitate ba ang attack ng porphyria when a patient go on binge drinking? Yes po. Ganon din po ang mechanism behind cytochrome P450 inducers. Siyempre, in-induce mo yung cytochrome P450, bababa din yung level ng heme, magiging overly active din ang ALAS. These are examples of drugs, no, that are unsafe, no, for patients with porphyria. Yung mga yan, cytochrome P450 inducers. Doc, di ko pa rin po ma-imagine yung pinagdidiscuss nyo, no? Litong-lito na po ako. Let's try to apply it. So for example, your patient has acute intermittent porphyria. Anong problem sa acute intermittent porphyria? You have a defect in porphobilinogen deaminase. Okay? Ngayon, yung patient, yung med student na taga section E, nag-binge drinking. Normally, di ba? Sorry. Normally, Kapag may defect ka sa enzyme, the intermediates before it would accumulate. Now, nag-binge drinking ka, alcohol, or na-depress ka, uminom ka ng cytochrome P450 inducers, kahit ano sa mga yon. What would happen again to the level of your heme? It will go down. What will happen to the inhibition of heme to your ALAS? It will be removed so that your ALAS becomes active. So madami ba siyang mapo-form na ALA and PBG? Yes po, Doc. Kaya lang, anong problema? May block na po dun sa pathway. Di siya makatawid. So tataas ng sobra ang ALA at PBG, thereby precipitating an attack of porphyria. Nasundan po? Clear po so far? May nagtataas ata ng kamay. Yes. Eric.
Yes, good morning, Doc. I just want to confirm. Tama po ba ang thinking ko when I say na ano po, CYP2E1 is a heme protein and that
Yes.
if you drink alcohol, hindi nakakayanan ni ADH. So yung heme that is supposed to inhibit tawag na to, ALAS as a negative regulator. ALAS is being, instead of being used to inhibit ALAS, is being used to make CYP2E1. Kaya nababawasan
Yes.
po ang level. Okay.
Yes.
Okay, thank you for clarifying, Doc.
Okay, sige. So, tapos na tayo sa iron, excessive alcohol, and CYP450 inducer. Let's look naman in fasting and glucagon. No? Pwede rin yan, no? Usually yung patient, no? Kunyari, anorexic yung patient, no? Ayaw niyang kumain, no? Tapos meron din pala siyang porphyria, no? Mape-precipitate din yung attack ng porphyria niya. Paano? Let's discuss first how we transcribe and translate ALAS. Paano tayo gumagawa ng ALAS? Okay. So first, when you go on fasting, ayaw mong kumain. So a hormone is then synthesized, no, or is then released, glucagon. Because we want to try to increase the blood glucose level kasi wala kang kinakain. Glucagon will bind to its receptor. Ang tawag doon, glucagon receptor. It is an example of a G protein coupled receptor, specifically a G stimulatory G protein coupled receptor. Pag G stimulatory ang G protein coupled receptor, it leads to an increase in cyclic AMP. Cyclic AMP will stimulate an enzyme, protein kinase A. The downstream effect of protein kinase A is to stimulate and allow the binding of CREB. Yung CREB is your COMP response element binding protein. It's a transcription factor. Magbabind siya in the CRE or COMP response element. Once CREB bind to CRE, it leads to the transcription and translation of a transcription factor, PGC1-alpha. PGC1-alpha is a transcription factor, and together with another transcription factor, FOXO1, would bind to the iron response element in the gene of ALAS. Since they are transcription factor, they stimulate the transcription of ALAS. Not shown here is your iron. Di ba sabi natin, iron is a positive regulator of ALAS. Kasi yung pagbabindan nila is an iron response element. So iron is a requirement there. PGC1-alpha, FOXO1, and iron, once they bind to iron response element, would stimulate transcription and translation of ALAS. So gagawa ka ng mRNA and eventually it will be translated to ALAS. In that sense, by fasting and with the use of glucagon, nakagawa ka ba ng maraming ALAS? Yes po. Dahil madami kang ALAS, ginadji ba uli yung pathway mo? Yes po. Kung may block ka, magpe-present ka ba or mape-precipitate ba ang attack ng porphyria? Yes po. Okay. So doon naman tayo sa other side. Sabi natin, presence of glucose as well as insulin can inhibit ALAS. Paano yon? When you take in glucose, anong hormone ang marirelease? insulin because we want to try to lower the blood glucose level to the normal levels. It will bind to its receptor, the insulin receptor, which is a receptor tyrosine kinase, no? It's an RTK receptor. The end effect of activation of receptor tyrosine kinase is phosphorylation of proteins. In this case, the one that's phosphorylated is your FOXO1. Nakita nyo, may phosphate. And phosphorylation of FOXO1 would render it inactive. Kapag inactive ang FOXO1, meron pa bang transcription factor na sasama kay PGC1 alpha and iron to transcribe and translate ALAS? Wala na po. Therefore, by giving glucose or insulin, can that decrease your ALAS? Yes po. That is why patients with porphyrias are given or are treated using glucose. Binibigyan po sila ng intravenous injection ng glucose as well as commercial preparation of heme known as your hematin. Okay? Naintindihan po how the regulators affect the synthesis of ALAS? Okay. Sabi nga natin, papunta pa lang po tayo sa exciting part. Guys, I really need your undivided attention no in the next few slides no kasi mas complex po siya. Medyo mas mahirap intindihin no but we'll try to simplify it. So let's now discuss the structure and function of your hemoglobin and myoglobin. So first, myoglobin. So myoglobin is a single polypeptide chain folded into 8 segments of alpha helices with designation of A to H. Okay? So pag tinanong kayo sa exam, what is the most common secondary structure that we can see in myoglobin? Sagot po, alpha helices. Okay? So it's mainly present in the heart and skeletal muscle that acts as an oxygen reservoir. That is one of its difference with your hemoglobin. Kasi di ba sabi natin when we discuss the heme proteins, hemoglobin is for transport of oxygen while hemoglobin acts as a reservoir in muscle that releases oxygen during active exercise. Okay? So the molecule myoglobin is lined up with polar amino acid on the exterior portion, no? Meaning yung mga water-soluble hydrophilic amino acid, and that the interior of the molecule is lined by non-polar amino acid with the exception of histidine. Protein chem. What is the polarity of your histidine? Ano uli yung polarity ng histidine?
Polar positive po.
Correct, no? It's polar basic or polar positively charged. Sabi nila, normally, sino ba yung sila? Sabi, no? Ang interior is normally non-polar or hydrophobic, no? Pero meron daw histidine residue that is polar doon sa interior. We have 2 histidine residue. One is in the distal segment, E7, that stabilizes the bond between oxygen and iron. Imagine niyo tong nasa center na to. Ito po ba yung heme na diniscuss natin kanina? Yes po. Sa gitna po ba niyan may iron? Yes po. Meron po bang oxygen na magbabind dyan? Yes po. The distal histidine stabilizes the bond between oxygen and iron. The other histidine is present on the proximal portion, F8, that directly binds with iron. Okay? So yun yung structure ng myoglobin. So tatandaan nyo, ang myoglobin, pag gumawa ka ng 4 niyan, no, it has a similar structure with your hemoglobin. Yung individual subunits, it's similar to myoglobin. Alright? So hemoglobin is a tetramer of 2 different types of globin chain with heme, ito yung nasa gitna, with heme acting as the prosthetic group. Okay? Therefore, depending no, on the stage of development or condition of the patient, that will determine the type of hemoglobin that he or she will have. Such that in adults, the major type of hemoglobin we see is hemoglobin A. It is made up of 2 alpha, and 2 beta chain. At tandaan nyo, laging dalawang klase, no, na nag-prepare. So hemoglobin is a metalloprotein that transports up to 4 molecules of oxygen. Now again, sabi natin, ang myoglobin for storage, oxygen for transport. And because of its complex structure, mas complex siya, di ba? Quaternary structure siya. It's able to transport other ions and molecules like hydrogen ions and carbon dioxide. Therefore, the reversible binding of oxygen to your hemoglobin is affected by allosteric effectors. Sa hemoglobin lang po yan nakikita. Wala pong allosteric effectors sa myoglobin. Pag sinabi nating allosteric effectors, these are the conditions that affects the reversible binding of your hemoglobin for oxygen. Siya lang po, no? Wala po si myoglobin. Ito pa, probably one of the more important slides. What are the bonds that holds your hemoglobin together? First, between your globin chains, alpha and beta chain, it is stabilized by hydrophobic interactions. Nakikita niyo po yung mga dots? Those are hydrophobic interaction, thereby creating 2 dimers, AB dimer 1 and AB dimer 2. Now, the bond that holds your dimer together, AB dimer 1 and AB dimer 2, are your hydrogen or ionic bonds. Nakikita niyo yung mga red na bonds? Those are hydrogen or ionic bonds. Ano pang other tawag natin sa ionic bond? Salt bridges. Salt bridges, no? So makikita niyo yan, pwedeng gamitin natin yan later on. Okay. Kasi importante yan, no, when we discuss the 2 different forms of hemoglobin. Meron kasing dalawang forms ang hemoglobin depending on its ability to bind and release oxygen. May T or taut form, R or relaxed form. Tandaan niyo po yung mga bonds na yan. So let's now discuss the globin chain synthesis, no? Na-discuss na natin yung heme synthesis. So dun tayo sa globin synthesis. So the synthesis of the globin portion of your hemoglobin is carried out by 2 gene cluster. First, your alpha globin gene family present in chromosome 16, responsible for encoding genes for transcription and translation of alpha chain, zeta chain, and pseudo alpha chain. The other one, the beta globin gene family, is located in chromosome 11, responsible for encoding genes for transcription and translation of beta and beta-like proteins or globins. Okay? Sabi natin kanina, ang hemoglobin, kailangan may dalawang globin chains, 2 different types of globin chains. Okay? In normal condition, it should come from alpha globin gene family, 2 should come from alpha, 2 should come from beta or beta globin gene family. Okay? Ano pa yung gusto kong emphasize dito? Next week, you'll be discussing thalassemia. Tama ba? Yes po, Doc. May reporters. So in the alpha globin gene family, tatandaan nyo, merong alpha and alpha 1 and alpha 2 gene. So pag tinanong ko kayo, how many genes encode for alpha chain in chromosome 16? Ilan po? Dok, masakit na sa ulo, VMAX na po. Di ba sabi natin ang chromosome natin laging pair, di ba? Pair of chromosome. So dun sa isang pair, meron kang alpha 1, alpha 2. Dun sa kabilang pair, may alpha 1 and meron ka ding alpha 2. Tama po ba? Yes po. So in total, how many genes? 4. Okay? Sinishare ko to sa inyo kasi nung med student ako, hindi ko talaga siya maintindihan. No? Ngayon na lang. Such that in alpha thalassemia, basta yun na yun. Alpha thalassemia, there are 4 types of alpha thalassemia depending on the gene that you will delete. Pag isa yung dinilit mo, ang tawag natin doon, silent carrier. Pag dalawa yung dinilit mo, alpha thalassemia minor. Pag tatlo, hemoglobin H disease. Pag apat, that's your hydrops fetalis. Iba naman siya when we're talking beta thalassemia, no? Basta may ang ano niyan, characteristic, beta 0, beta plus, and B. Ito normal, ito reduced production, ito completely absent. Iwanan ko na po yan sa mga reporters. So let us discuss now, sabi ko di ba kanina, depending on the stage of development, that will determine what type of hemoglobin you'll have. So hemoglobin synthesis begins in the first few weeks of embryonic development within the yolk sac, where the major type of hemoglobin you produce is your hemoglobin goer 1, made up of zeta chain coming from the alpha globin gene family and epsilon coming from the beta globin gene family. Nasunod po ba yung dalawa galing sa alpha, dalawa galing sa beta? Yes po. The other types of hemoglobin, hemoglobin goer 2 and Portland. By 6 to 8 weeks of gestation, the expression of these types of hemoglobin dramatically declines, coinciding with the change of hemoglobin synthesis from the yolk sac to the liver. Kasi natutunan nyo sa embryology that yolk sac is only a temporary structure. Tama po ba? Yes po, Doc. At around 10 to 12 weeks of gestation, we form another type of hemoglobin, that is your hemoglobin F or fetal hemoglobin, that has two types of globin chain. Alpha from the alpha globin gene family and gamma from the beta globin gene family, coinciding with the dramatic decrease in the epsilon and zeta. Ito na po yun, alpha and gamma. Shortly before birth, bago ipanganak yung baby, as you can see, there is a smooth switch in the expression of your gamma globin and an increase in your beta globin. Kakaroon ng switch. So that before birth, we begin producing your adult type of hemoglobin, hemoglobin A, with alpha and beta chain, alpha from alpha, beta from beta globin gene family. Right before birth, ano pang napansin nyo? There's another type of globin chain that you produce. That's your delta chain. So we can also produce hemoglobin A2, which is composed of alpha from the alpha globin gene family and delta from the beta globin gene family. That's why if you're going to be asked, what are the hemoglobins that we can see at birth? Ito pong tatlo. Hemoglobin F, hemoglobin A, and hemoglobin A2. Ito din po yon. So again, what is hemoglobin A? Made up of alpha and beta, present in 96% of adults, 25% in newborn. Hemoglobin A1C has the same globin chain component of hemoglobin A. It's just that the terminal valine residue, kita nyo, would react with glucose in a process called as your glycation. Glycation, that is different from glycosylation. Glycation is a non-enzymatic process. The rate of glycation depends on the concentration of glucose. So pag mas mataas ang glucose concentration mo, mas mataas ba ang hemoglobin A1C mo? Yes po, Doc. The normal hemoglobin A1C should only be around 5%. That is why we can use hemoglobin A1C to diagnose diabetes mellitus. And a value of more than or equal to 6.5% would tell you that the patient has DM. But there's again an exception to the rule. In patients who lose their blood prematurely, sino ba yon? Yung may mga hemolytic anemia, hemolysis. Ano ang value ng hemoglobin A1C? It's falsely low. So we cannot use hemoglobin A1C as a diagnostic tool in patients with hemolytic anemia. Okay? Please remember that. Hemoglobin A2 naman is composed of alpha and delta chain, 2 to 3% in adult, but is higher in patients with beta thalassemia. In beta thalassemia, you have problems producing the beta chain. Ano nga uli ang pinakamadaming hemoglobin in adults? Hemoglobin A, which has hemoglobin B, di ba? Or globin or beta globin chain. So since you have a problem producing beta chain, pwede bang mag-associate ang beta-like chain with alpha? Pwede din po. Kaya po tumataas ang hemoglobin A2 in patients with beta thalassemia. And lastly, you have your fetal hemoglobin made up of alpha and gamma chain, 75% in newborns. No, and please take note, the important characteristic of your fetal hemoglobin is that it has a higher affinity for oxygen than hemoglobin A. And that is beneficial for fetus, no? Kunyari, inside the uterus, no? That is beneficial for them so that the maternal RBC containing hemoglobin A, when it traverse or when it reach the placenta and naka-encounter niya sa kabilang side, sa fetal side, yung fetal hemoglobin, which has a higher affinity for oxygen, the fetal hemoglobin. So anong tendency? The oxygen from the maternal side will then transfer to the fetal side. Okay? Kasi hindi pa naman sila nakakahinga on their own, no? So they depend, no, on the oxygen from the maternal side. Any questions up until this point? Questions? Sige. Gusto niyo ba ng break? Sige. Break, bladder break, doc. VMAX na po talaga. VMAX, saturated na po. Sige, okay. Let's have a 3-minute break. Alright, resume ko lang po yung recording. Okay, so for the next part of our discussion, we'll be discussing oxygen binding to hemoglobin and myoglobin. So there are actually two forms of hemoglobin depending on its ability to bind oxygen, no? First, we have your T or taut form. That is also called the low oxygen affinity form. In the deoxyhemoglobin form, meaning walang oxygen na nakakabit doon sa hemoglobin, no? The ionic, no, or polar bonds are said to be multiple and they are present in increased number. Therefore, your hemoglobin is in a rigid position, hindi siya makagalaw. That is what we call the T or taut form or the low oxygen affinity form. Imagine it like this female, no, with her arm wrapped around her chest. Ayaw niya mag-accept ng oxygen, no, does not want to accept oxygen. So the tendency, if the hemoglobin is in the taut or T form or tense form, is to release oxygen. At higher partial pressure, the tendency is for the first oxygen to bind to the first subunit, disrupting some of those ionic bonds. Itong mga to, it will disrupt or remove some of the ionic bonds, affecting the other subunits, making it easier for the second oxygen to bind. Binding of the second oxygen molecule would again disrupt some of those ionic bonds, some of those salt bridges, no, allowing the third oxygen to bind, no, easier than the second oxygen. And lastly, ang pinakamabilis mag-bind sa hemoglobin is the last oxygen molecule. Again, that is due to the disruption of the ionic or polar bond. Imagine it like this kid with his arm wide open, ready to accept oxygen. So that binding of oxygen, no, in your hemoglobin, disrupting those ionic or polar bonds or salt bridges, converts, no, the hemoglobin from the taut or tense form or T form, the very rigid form, to your relaxed form or the high oxygen affinity form. Okay? So pag na-disrupt yung mga ionic bonds, yung salt bridges, that converts your hemoglobin into a relaxed, mute ko lang, ayan. that converts your hemoglobin into its relaxed, no, or R form, the high oxygen affinity form. The property of hemoglobin, yung property niya, no, that makes it easier for succeeding oxygen to bind, no, is what we call your cooperative binding or positive cooperativity effect. Imagine it like this. For example, this is your heme. Diba? Ito yung heme. Para kang naka-side view sa kanya. This is the plane of your heme. What can you notice about the iron at the center of the heme? It is located somewhat lower in comparison to the plane of your heme. In the presence of oxygen, nakita nyo? Oxyhemoglobin. What happens to the iron? It lifts, no, the iron at the same plane of your heme. What is the end effect? Is the disruption of your ionic pair. Pag nagdagdag ka ng oxygen, it will disrupt the ionic pair, making it easier for the second oxygen to bind. When the second oxygen binds, again, it will disrupt the ionic pair, making it easier for the third oxygen to bind, and again, disrupt some of those ionic pair, making the last oxygen have the easiest time, no, of binding to the last subunit. Again, we call that the cooperative binding of your hemoglobin or the positive cooperativity effect. Let's now go through your oxygen dissociation curve. Oxygen dissociation curve plots the percent saturation on the y-axis versus the partial pressure of oxygen on the x-axis and its contribution to the total oxygen content. Myoglobin and hemoglobin has its own oxygen dissociation curve, which you'll see on the next slide. Let's try to define some terms. P50, what is P50? P50 represents the partial pressure at which hemoglobin or myoglobin is 50% saturated with oxygen. To put it in perspective, para din po yung nasa enzyme nyo sa Michaelis-Menten curve. Sino ang katumbas niya dun sa Michaelis-Menten curve?
Yung KM po.
Correct, no? Your KM. Di ba? Ano nga uli ang definition natin ng KM? KM is the substrate concentration in which your enzyme is 50% saturated. Tama ba? Yun ba yung KM? Di ba? So, ganun din dito. So, P50 is the concentration of or your partial pressure of oxygen in which your hemoglobin and myoglobin is 50% saturated with oxygen. So, pwede din po ba nating i-apply dyan ang characteristics ng KM? Yes po. Doc, sabi po ni Doc Bravo, KM has an inverse relationship with the affinity of substrate to the enzyme. Ganon din po ba dito? Yes po. P50 relates the affinity of oxygen now to your hemoglobin or myoglobin. Baliktad lang po. Pag mababa ang P50, ano ang affinity ng oxygen to hemoglobin or myoglobin? Mataas. When you have high P50, ano naman po ang implication nun? Mababa ang affinity ng oxygen to hemoglobin and myoglobin. Nasusundan po? Clear? Kasi mas mahirap siyang kung hindi natin i-apply doon sa same same principle.
Doc, pwede pong isa pa po.
Sige, okay. Sabi natin, ang P50 is the concentration of partial pressure of oxygen in which your hemoglobin or myoglobin is 50% saturated with oxygen. Applying it to enzyme kinetics, that is also like the KM. KM is the substrate concentration at which your enzyme is 50% saturated with substrate. Diba? Doon sa Bmax 1/2, kung ano yung substrate concentration doon, yun yung KM. So ang characteristic ng KM can also be applied to P50. That is inverse relationship with the affinity of substrate or oxygen to enzyme or hemoglobin and myoglobin respectively. O, apply natin, kunyari dun sa enzyme kinetics, balikan. High Km, ano ang affinity ng substrate for the enzyme? High Km. Since inverse ang relationship niya, low. So pag low ang Km, high affinity of substrate for the enzyme. Pag high ang Km, low ang affinity ng substrate for the enzyme. Ganon din dito. Pag low ang P50, ano ang affinity ng oxygen to your hemoglobin or myoglobin? Mataas. Pag high ang P50, anong affinity ng oxygen kay hemoglobin or myoglobin? Mababa. Later on, ma-apply niyo yan when we discuss the right shift or left shift of your hemoglobin dissociation curve. So, oxygen saturation, that measures the percentage of hemoglobin binding sites occupied by oxygen sa y-axis. And the partial pressure of oxygen is the pressure of oxygen in a mixture of gases, usually 160 millimeter mercury at sea level. All right, so let's now compare the oxygen dissociation curve of myoglobin and hemoglobin. Hindi ko na po kayo pahihirapan. I just like you to know the characteristic or yung itsura ng hemoglobin and myoglobin oxygen dissociation curve. Excuse me. Okay, so on your left is your myoglobin dissociation curve. What can you notice? It rises sharply, di ba? It rises sharply and then levels off, resembling a hyperbolic curve. Di ba? Parang hyperbolic yung shape niya. The P50, sabi natin, the P50, which reflects the binding sites of your myoglobin being 50% saturated, is relatively low, mababa, 1 to 2 millimeter mercury or 1 to 2 torr. That tells you, kapag mababa ang P50, what is the affinity of oxygen to myoglobin? Mababang P50.
Very high affinity po.
Mataas po.
High affinity. Okay, so that will tell you that oxygen has a high affinity to myoglobin. Therefore, myoglobin is ineffective in transporting oxygen. Rather, its main function is to store and release oxygen during strenuous exercise. Imagine niyo kung sa hemoglobin nyo, no, or sa RBC nyo, ang nandoon myoglobin, no? Yung tissues ninyo, gustong-gusto na ng oxygen. They are actively metabolizing. I-release ba ng myoglobin yung oxygen na hawak-hawak niya? Hindi niya basta-basta i-release because of its high affinity. Okay? That is because it has a low P50. On the other hand, the hemoglobin oxygen binding curve exhibits special features. First, it has a sigmoidal shape, no, suggesting that binding of oxygen at one site increases the chance that the oxygen would bind to the remaining unoccupied site. Oh, anong tawag natin uli doon? That is the cooperative binding or the positive cooperativity effect. And that results in structural changes. Sabi natin, di ba? What is the structural change that we see from the T or taut form to the R or relaxed form? The disruption of ionic bonds of the salt bridges. We can see that as the steep upward curve in the region near 20 to 30 millimeter mercury. Pansinin nyo, at 25% saturation dito, no? Anong kailangan mong P50 just to saturate the first binding site? Maybe around 18 to 20 millimeter mercury. Pero dun sa P50, no, you only need to add a few partial pressure of oxygen just to saturate the second binding site. Kaya nakikita nyo, very steep yung pag-akyat niya. That tells you na may nangyari, no, dun sa first binding ng oxygen sa hemoglobin, kaya konting increase na lang sa partial pressure that would make it easier for the second oxygen to bind to the second binding site. Okay? So that is your positive cooperative binding. Now, let's discuss what are the factors that affects the reversible binding of oxygen to your hemoglobin. Ang tawag natin doon are the allosteric effectors. Balikan natin, sabi natin, allosteric effectors only applies to hemoglobin. Tama po ba? Nag-a-apply po ba siya sa myoglobin? Hindi po. Okay? Sa hemoglobin lang po. So what are the conditions that affects the reversible binding of oxygen to hemoglobin? Ito pong mga ito. PO2, partial pressure of oxygen, partial pressure of carbon dioxide, pH, 2,3 BPG, and temperature. Okay, mag-alala, iisa-isahin natin yan mamaya. Such that a right shift, pag nag-shift yung curve to the right, yung black line dyan, guys, on the right, no, that is your normal hemoglobin oxygen dissociation curve. When the curve shifts to the right, as you can see here, from here to here, ano nga uli yung P50? That is the partial pressure at which your hemoglobin is 50% saturated. Pag nag-shift to the right, what happened to the P50? Tumaas po ba? Bumaba? Nag-increase po ba yung value? Tumaas. So that tells you that you need a higher partial pressure of oxygen just to saturate 50% of your hemoglobin. So anong ibig sabihin nun? Mas mataas ba ang affinity ng oxygen to hemoglobin or mas mababa? Mababa. That is low oxygen affinity. Ano nga uli yung form ng hemoglobin that is the low oxygen affinity? T or R?
T-form.
T-form. T-form, the tense form. Naalala nyo kanina, siya yung mas rigid. Siya yung babae na naka-close yung hand sa arms. So ano ang tendency ng T-form? To accept or to release oxygen?
Release.
To release oxygen. These are the conditions that shifts the curve to the right. These are the conditions that you see in the peripheral tissues, no, where cells are actively metabolizing. They have higher PCO2, higher partial pressure of carbon dioxide, high hydrogen concentration, high 2,3 BPG, high temperature, isama na po natin, low partial pressure of oxygen, or low pH. This again are the conditions seen in the peripheral tissues. Is it beneficial for actively metabolizing tissue na ang form ng hemoglobin na dadaan sa kanya is in the low oxygen affinity form? Aba, yes, Doc. Kasi they are actively metabolizing. Therefore, do they need oxygen? Yes po. Pagdaan ng 10 state, tout state, or T state doon sa peripheral tissues, actively metabolizing, i-release niya ba yung oxygen niya? Yes po. Alright? So that's the right shift. On the other hand, no, a left shift, pag sa kabila naman, no, anong na-notice niyo sa P50? Ano, high or low?
Low po.
Low, Doc.
Lower, no? Bumaba yung P50, no? So sabi natin, pag low P50, ano uli ang affinity ng oxygen to your hemoglobin?
High po.
Inverse ang relationship. High affinity. So sa left shift, what form of hemoglobin predominates? The R form or the T form?
R form, Doc.
The R form. So ano ang tendency since it's the high oxygen affinity form? To accept oxygen or to release oxygen? Okay, and these are the conditions that shifts the curve to the left. These conditions are usually seen in the lungs, no? Where you load hemoglobin with oxygen. Baliktarin nyo lang po, kabaliktaran lang nito. Low PCO2, low hydrogen concentration, low 2,3 BPG, low temperature, high PO2, or high pH. Nasundan po? Okay. So sa next slide, we'll be discussing how, no, can the allosteric effectors affects, no, the transition of your hemoglobin from T to R form or from R to T form. Sige, wait lang po guys ha. I-pause ko lang saglit kasi may nagda-doorbell. Okay, I'm back. Sige. So let's now discuss, no, the different allosteric effectors. First, pH. Carbon dioxide. Sama na natin siya ng hydrogen concentration. Excuse me. So sabi dito, hydrogen ions facilitates reformation of salt bridges. Naalala nyo yung salt bridges? ionic bonds. Ano nga uli yung ini-stabilize niya? The AB dimers. Mas madaming salt bridges, mas madaming ionic interaction. What form of hemoglobin will predominate? Is it the T or R form? Mas madaming salt
The
bridges, mas rigid siya.
T form.
T4.
The T form. And since it's in the T form, ano ang tendency ng form na yon? To release or accept oxygen? Ito naman, nandito. That is to release oxygen. Sabi natin kanina, actively metabolizing tissue would produce a lot of hydrogen ions, would also produce a lot of carbon dioxide. Tama ba? And product ba yan ng actively metabolizing tissues? Yes po, Doc. No? So, makikita nyo that carbon dioxide, no, produced by actively metabolizing tissue from the Krebs cycle can actually form carbonic acid by the enzyme carbonic anhydrase, converting it now to bicarbonate and hydrogen ions that can be buffered by the hemoglobin, reforming now the salt bridges, facilitating now the TENS, the T, or the tau form. Crystal? I-mute lang. Hindi ko ma-mute si ayan. So again, reformation of salt bridges by high levels of hydrogen ions facilitates the conversion of hemoglobin to the TENS, T, or tau form. Okay? So carbamates naman, ano ba yung carbamates, no? These are produced by the reaction of carbon dioxide, no, with the amino group in your hemoglobin producing carbamates. Ito po yung carbamates. Okay? In the process, ano yung na-produce nyo? Hydrogen ions. Can that facilitate or can that reform the salt bridges? Yes po, Doc. Can that facilitate the formation of TENS or T-state? Yes po. Ano nga uling form yon? Is it the high or low oxygen affinity form? The low oxygen affinity form. Releasing oxygen. Oh. Anong manonotice nyo dito? With decreasing pH, ano nangyayari sa curve? It shifts it to the right. Diba sabi natin, right shift, no, converts it to the low oxygen affinity form, releasing the oxygen. Okay? Low pH or high hydrogen concentration. Pwede din. Okay? Next, temperature. Di ko na po kayo pahirapan, no? Temperature has an inverse relationship with your oxygen affinity. The higher the temperature, the lower the oxygen affinity, the lower the oxygen affinity, or the lower the temperature, the higher the temperature. Next, 2,3-BPG. 2,3-BPG is actually a product of the glycolytic pathway, glycolysis, no? So yung 1,3-bisphosphoglycerate sa glycolysis can be converted to 2,3-BPG. Ano bang meron sa 2,3-BPG? It's an anionic molecule, meaning it's negatively charged, that can associate itself at the center of your hemoglobin, specifically in the beta chains, no? In the beta chains, forming now salt bridges or ionic bonds between these amino acids. So gumawa siya ng salt bridges. Mas marami bang ionic bonds o salt bridges? Yes po. Which form will predominate? The T or R form?
T.
T
The T form. High oxygen affinity or low oxygen affinity?
Low, Doc.
Low oxygen affinity. Ako, iniisip ko lang yan nung med student ako. Pag actively metabolizing ang cell or ang tissue, active din ba yung glycolysis niya? Yes po, Doc. Kasi glucose, no, is the substrate for glycolysis, no? So, ganun din po. Lastly, carbon monoxide, di po natin yan diniscuss dun sa allosteric effector, but it can also affect your hemoglobin dissociation curve. It is an odorless and colorless gas that can bind on the same site, no, for oxygen in hemoglobin. Kung nagbabind siya dun sa same site, masasaturate mo pa ba fully ang hemoglobin? Hindi na po. So, anong tendency ng curve? Shifts it downward. Okay? Ano pang na-notice nyo? Yung black yung normal. Doc, bakit po nag-shift to the left? Ano po yung shift to the left? High oxygen affinity form or low oxygen affinity form? Doc, that's the high oxygen affinity form. Eh sabi mo, Doc, mas mataas ang affinity ng carbon monoxide with oxygen. Bakit ngayon tataas yung oxygen affinity form? That is because, di ba, you have 4 subunits. Once carbon monoxide binds to one of the subunit, it will not allow unloading of the remaining oxygen, increasing its affinity. So pag tinanong kayo sa exam, what is the effect of carbon monoxide to your oxygen hemoglobin dissociation curve? Downward and leftward. Okay? Yan, parang ganyan. Meron na, naghahanap pa ng iba. So let's now go to your hemoglobinopathies, no? So hemoglobinopathies are heritable mutations that affects the expression of globins, the amino acid sequence for globins, or both, no? Ang i-discuss ko lang po in the succeeding slide will be your hemoglobin S and hemoglobin C, kasi yung isang hemoglobinopathy, thalassemia, I will leave that for the reporters. Okay? So first is your hemoglobin S or sickle cell disease. This is an autosomal recessive disorder caused by a point mutation on a single nucleotide leading to substitution of valine for glutamic acid at position 6 of the beta globin chain. Dok, ang dami namang information dun sa isang sentence na yon. It only tells us that there is a change in amino acid from the original glutamic acid to valine. Okay? So it's an amino acid substitution. Napalitan yung amino acid. This is a form of a polar to non-polar. No? Polar to non-polar amino acid substitution. Okay? So ano yon? Ano bang polarity ng glutamic acid?
Polar, Doc.
So, okay, it's polar. Ano yung charge?
Negative.
Negative.
Polar, negatively charged. While valine, ano ang polarity niya? It's non-polar. Okay? What's the sequelae? Anong effect niya, no? When hemoglobin, no, with a mutation becomes deoxygenated, no? We call that deoxyhemoglobin S. You form sticky patch, nakita niyo to, in the beta globin chain. Kasi nga ang problem lies in the beta globin that can associate on a crevice present in your deoxyhemoglobin A. So kapag ang hemoglobin is in the deoxy form, they can polymerize and cause the sickling of your RBC. Magsisickling sila. And because of this sickling, no, we can observe the different clinical sequelae of your sickle cell disease. Kasi ang sickled RBC, it's not pliable, no? Unlike the normal biconcave RBC, very pliable. So pag dumaan yan sa capillaries, no, especially in the sinusoids of your spleen, matatrap na sila doon, no? Mamamatay na sila doon, maghihemolyze na sila doon, no? So depending on the organ where it will lodge, no, or matatrap, no, that will give you the clinical symptom. For example, sa brain, then you can have stroke-like symptoms, no, or sa heart, you can have myocardial infarction or acute chest syndrome. Okay? Hemoglobin C disease naman, same amino acid, same position, sa 6th position pa din, sa beta globin gene pa din, sa beta globin chain. This time, you have a glutamate to lysine substitution. That is a form of a polar to polar substitution. Ano yon? Anong polarity ng glutamic acid? Polar, negatively charged. How about lysine? What's the polarity?
Positive.
Polar positive.
Okay, polar positive, no? Kapatiran niya ng arginine, histidine, no? Yan ang mga positively charged. So unlike hemoglobin S or sickle cell disease, this patient would not suffer from sickling kasi mas less yung severity if it's a polar to polar substitution. But they will have mild chronic hemolytic anemia. On your right, you'll see the original glutamic acid residue in hemoglobin A, valine, no? That is replaced, no, in hemoglobin S, and lysine in hemoglobin C. Such that if you subject them in electrophoresis, this is the cathode or negative side, anode, positive side. Which hemoglobin will travel the farthest? That would be your hemoglobin A because of its glutamic acid residue that is negatively charged, more attracted on the anode side, followed by hemoglobin S containing non-polar amino acid, and hemoglobin C containing lysine, which is polar, positively charged, more attracted on the anode side. Okay? Clear po? Okay. Any questions up until this point? Sige, so we're done with porphyrins, heme synthesis, myoglobin and hemoglobin structure and function. Hemoglobinopathy. So let's now go with your heme degradation. Okay. So kanina 'di ba sabi natin yung mga heme proteins, they contain proteins and heme, no? So ano-ano ba yung mga sources natin ng heme? Pwedeng sa age RBC after 120 days, from myoglobin when you have muscle damage, no, or cytochromes, no, from your liver, cytochrome P450, cytochrome C, and other enzymes containing heme. So the iron and the globin portion, no, can be recycled, no. Natutunan niyo 'yan doon sa nitrogen balance niyo, 'di ba? Yung proteins can be recycled to join the amino acid pool, no. That is also true for iron, pwede siyang ma-recycle. While the heme portion should be degraded, no. Ang end product ng heme degradation is your bilirubin. So ang discussion natin dito would be this portion, how heme is degraded. First, what is the enzyme that would initially degrade your heme? So that is a microsomal enzyme called heme oxygenase, seen in the reticuloendothelial system, commonly in the macrophages, no? Macrophages. Presence of heme induces the activity of heme oxygenase, the rate-limiting enzyme for heme degradation. Kabaliktaran naman po ang effect ng heme doon sa heme synthesis, di ba? Naalala nyo? Anong effect ng heme sa alas? It negatively inhibits alas. Dito, it stimulates the rate-limiting enzyme that degrades your heme. So heme oxygenase is a NADPH oxygen-requiring enzyme that would cleave the pyrrole rings, the methane bridge. Naalala nyo kanina, how do we label the methane bridge? Using Greek alphabets, alpha through delta. Specifically, heme oxygenase would cleave the alpha methane bridge. Kita nyo? Converting your heme to a green pigment, biliverdin. Okay? So this is the enzyme. So what are the end products of your heme oxygenase? Biliverdin, the green pigment, carbon monoxide, no, which is by the way, heme oxygenase is the only enzyme in our human body capable of producing carbon monoxide. Pero ano yan, it's not insufficient amount to cause toxicity, no. And lastly, you have ferriiron. Okay, so yung tatlo after heme oxygenase. Now, your biliverdin would be furtherly reduced to the yellow pigment bilirubin with the help of the enzyme biliverdin reductase, which is a NADH or NADPH requiring enzyme. Yan. So it's also NADH or NADPH dependent. So from biliverdin to bilirubin, the yellow pigment. Please remember that the initial bilirubin that you produce is a lipophilic molecule. Kaya naman, the initial bilirubin that we produce comes in several names. It can be water insoluble. Pwede rin niyang lipid soluble. Yun din po yun. Pwede rin po yung tawaging unconjugated bilirubin. Indirect bilirubin or B1. Lahat po yan pertains to the initial bilirubin that we produce. Now, being lipophilic, produced by the reticuloendothelial system, tatandaan nyo, ang ultimate place where bilirubin will be conjugated is in the liver. So, kailangan ba niyang makapunta from the macrophages to the liver? Yes po. Anong problem? This is lipophilic. Our circulation, the plasma, is said to be aqueous environment. Polar. Polar. So ito, non-polar. So makakatawid ba siya sa circulation basta-basta? Hindi po. That is why it needs a carrier in the form of albumin. Okay? Albumin, the carrier of bilirubin, the unconjugated one. Albumin has two binding sites for bilirubin, no? It has a high affinity binding site and a low affinity binding site. What I would like to focus on is in the low affinity binding site. Bakit? Because low affinity binding site binds your bilirubin loosely, mabilis matanggal, such that presence of drugs, for example, sulfonamides, salicylates, tetraioxone, and warfarin can displace the bilirubin from the loose binding site. And pag na-unbound yung bilirubin, unconjugated bilirubin, lipophilic, saan ang tendency niyang pumunta? Sa areas na lipophilic din. And that is in your brain. It can deposit in the brain, causing damage to the neurons in a condition called as kernicterus. Okay? That is why premature babies, no, where their liver is still premature, we don't give these drugs. Okay? Because that can displace, pwede niyang elbowin yung bilirubin sa loose binding site, pumasok sa brain, and cause kernicterus. Okay? So let's now go back to your albumin-bilirubin complex. Ito na po siya. Okay? The albumin and bilirubin complex would go inside the hepatocyte in a carrier-mediated, facilitated diffusion. Sabi sa Pisho, ano sabi ni Doc Barbon? Pag facilitated diffusion, energy dependent, energy independent. It's energy independent. Is it concentration dependent? It's concentration dependent. Such that kapag ang bilirubin inside the hepatocyte is already high, papasok pa po ba ang bilirubin from the circulation? Hindi na po. Okay? So what will happen? Nakita nyo, iniwanan na siya ni albumin and it goes inside the hepatocyte. Being lipophilic, its tendency is to efflux out of the hepatocyte. But inside the hepatocyte, we have proteins, ligandin, otherwise called as glutathione S transferase and protein Y that will keep your bilirubin soluble, preventing it to efflux out of the hepatocyte or the bloodstream. Nasusundan? Ganito siya. Imagine niyo ganito siya. From the portal vein or hepatic artery, papasok siya sa hepatocyte. Sa hepatocyte, mako-conjugate siya, lalabas siya sa bile canaliculi. Ganun po ang flow ng metabolism ng heme, no, to bilirubin. So sige, na-imagine nyo na? Let's proceed. So what happens inside the hepatocyte is that the bilirubin would undergo 2 rounds of glucuronidation using UDP glucuronate or UDP glucuronic acid. Being a lipophilic compound, pag kinabitan mo siya ng acid, no, magiging polar na ba siya? Yes, Doc, kinabitan natin ng acid. 2 glucuronic acid. The enzyme that will do that is UDP, glucuronyl transferase, or UDP, glucuronyl transferase. Parehas lang po yun. Okay? It will now convert your bilirubin into a water-soluble bilirubin called bilirubin diglucuronide. Marami siyang name. Water-soluble, lipid-insoluble, B2, conjugated bilirubin, or direct bilirubin. Pare-parehas lang po. Alright? So this picture just shows you that we need 2 molecules of glucuronic acid to form your bilirubin diglucuronide using the enzyme UDP glucuronosyl or glucuronyl transferase. Okay? Now, meron na tayong bilirubin diglucuronide here. So what is the fate of your bilirubin diglucuronide? It can be transported in an active energy-dependent process inside, no, here in your MRP2 or multi-drug resistance like protein 2, otherwise called as ABC, or MOAT, multi-specific organic anion transporter. Going now to the bile canaliculi. Naalala nyo kanina sa picture from the hepatocyte to the bile canaliculi. Or some of the conjugated bilirubin can efflux out of the hepatocyte only to be taken back by this transporter o ATP 1B1 and 1B3. Dok, may importansya ba yung pinagsasabi mo? Ang dami naman yung transporter na yan. Yes po. Later on, when we discuss the different hyperbilirubinemias, makikita niyo po sila. Okay? Now, what happens? Once secreted in the bile canaliculi, ang ultimate location na pagpupuntahan niya is in the intestine. In the intestine, we have normal flora that secretes enzyme like beta-glucuronidases that liberates the bilirubin. Yung kinabit na glucuronic acid will remove it to liberate bilirubin. This enzyme also transports or converts bilirubin to a colorless molecule called urobilinogen. Iba to dun sa mga porphobilinogen, coproporphyrinogen, no? Iba po, urobilinogen. What happens to your urobilinogen? Urobilinogen can undergo enterohepatic circulation. Pwede siyang bumalik sa liver. Pwede siyang matransport sa kidneys in the urine in the form of urobilin, which gives color to your urine. But majority of your urobilinogen is converted to stercobilin, which is the one that gives color to your feces. Ito din po yun. Initially, heme is degraded in the reticuloendothelial system, forming unconjugated bilirubin, indirect bilirubin, or water-insoluble bilirubin. Being lipophilic, kailangan ba niya ng transporter? Yes po, Doc, in the form of albumin. It will then go to the liver where it undergo 2 rounds of glucuronidation with the help of the enzyme UDP glucuronyl transferase. We now have conjugated bilirubin or bilirubin diglucuronide, otherwise called as direct bilirubin, water-soluble bilirubin that is transported into your bile canaliculi going into your intestine. In the intestine, no, enzymes from the gut bacteria would liberate the bilirubin, converting it to your urobilinogen, where some of it can undergo enterohepatic circulation or in the kidneys as urobilin, but majority is excreted in the feces as the form of stercobilin. All right? Any questions? Clear so far? Ito, pagod na po kami makinig. Sige. Trust me guys, mas nakakapagod pong magsalita. Sige. So last is the disorders of bilirubin metabolism, no? So tatandaan nyo, the normal serum bilirubin in adults and in children should be less than 1 mg per dl. Pag lumampas, pag tumungtong na yan ng 1, ang tawag natin doon, hyperbilirubinemia. And that is different from jaundice. Tatandaan nyo, hyperbilirubinemia and jaundice are 2 different things. Diba? Jaundice being a physical examination finding. No? It's an objective finding of hyperbilirubinemia, and you can see it when bilirubin is approximately 2 mg per dl. Saan ba galing yung hyperbilirubinemia, Doc? So there are reasons, no, or there are causes of hyperbilirubinemia. First, excess production of bilirubin more than the liver can conjugate. Most commonly, when RBCs hemolyze. Pag masyadong maraming RBC ang namamatay, no? Marami ka bang mapo-form na bilirubin unconjugated? Yes po, that can be overwhelming for the liver to conjugate. Or from the inability of the damaged liver, no, to excrete bilirubin. Mismo yung liver mo ang may problem, no? The problem in conjugation, no, of your bilirubin. Or after may conjugate ng liver, you have obstruction in the biliary tree that prevents excretion of conjugated bilirubin. Such that we can categorize also hyperbilirubinemia as prehepatic, hepatic, or post-hepatic. In prehepatic, this is mainly due to hemolytic anemias. Pinaka-common po for biochemistry, sickle cell anemia. We've already discussed this a while ago. Pyruvate kinase deficiency or G6PD. Okay? Next, hepatic, anong mag-elevate when you have prehepatic prehepatic jaundice? That is your unconjugated bilirubin. Okay? Unconjugated bilirubin. In hepatic hyperbilirubinemia, no, the problem is in the liver, no? It can be due to neonatal or physiologic jaundice or inherited conditions of the enzymes that conjugates the bilirubin. Yung UDP, glucuronidase transferase, no? Ito pong mga condition na ito. Crigler-Najjar has two types, type 1 and type 2. Gilbert syndrome also affects UDP, glucuronidase transferase. So what type of hyperbilirubinemia would elevate in these two conditions? Unconjugated pa din, no? Kasi the problem is in the enzyme that conjugates your bilirubin. And lastly, post-hepatic, no, can be due to obstruction, no? So that's self-explanatory, no? Na-conjugate na, no? Na-conjugate na, hindi lang ma-excrete due to obstruction. That can be due to stones, no, in your biliary tree or mass, no, may bukol, no, in the biliary tree. Or parasites, pwede din yon. Pag nag-lodge yung parasite dun sa biliary tree, hindi makalabas. That can cause conjugated hyperbilirubinemia. Dubin-Johnson syndrome is a problem in the MRP2 transporter. Nakita na ba natin to? Yes, Doc. After makonjugate ng bilirubin, no? That the transporter before the bile canaliculi is the MRP2 transporter. When this is defective, na-conjugate mo na ba yung bilirubin? Yes po, Doc. So ano mag-elevate sa'yo? Conjugated bilirubin. The other one is Rotor syndrome. Rotor syndrome is a problem in OATP 1B1 and 1B3. Na-encounter na ba natin to? Yes po, Doc. This is the transporter that brings back conjugated bilirubin inside the hepatocyte. So anong bilirubin ang mag-elevate dito? Conjugated, unconjugated? Conjugated hyperbilirubinemia. Okay? As a side note, Rotor syndrome is actually discovered by a Filipino doctor, no, Filipino internist, no, si Dr. Rotor. So we can also categorize hyperbilirubinemia as unconjugated or conjugated. Ayaw niyong gamitin yung pre-hepatic and post-hepatic, pwede rin conjugated, unconjugated. Hemolytic anemia, sabi naman na natin, pag maraming RBC ang na-hemolyze, you'll have more bilirubin that the liver cannot conjugate, no? So you'll have unconjugated hyperbilirubinemia, physiologic jaundice, Crigler-Najjar and Gilbert syndrome, problems in UDP, glucuronidase transferase, and toxic hyperbilirubinemia. So for conjugated hyperbilirubinemia, that's obstruction in the biliary tree, Dubin-Johnson syndrome, a problem in MRP2 transporter, Rotor syndrome, a problem in your OATP transporter, no, or liver disease, no, like in hepatitis. Okay, so I think that's the last slide for my lecture. Okay.